Hello everyone, my name is Eduardo Lamas, and in this video we will talk about phagocytes and their main functions. Innate cells include phagocytes and non-phagocytes. Among the phagocytes, we have granulocytes, which include all the phils, eosinophil, neutrophils, and basophils. However, out of these three, only neutrophils are phagocytes. The main function of neutrophils is phagocytosis and activation of bactericidal mechanisms. And other phagocytes are macrophages, which are derived from monocytes. Macrophages also participate in phagocytosis and in the activation of bactericidal mechanisms. In addition, macrophages are antigen-presenting cells. A third class of phagocytes are dendritic cells, which include conventional dendritic cells that participate in the T-cell activation of the adaptive immune response, and plasmacytoid dendritic cells, which participate in the antiviral response by releasing massive amounts of interferon. Hematopoiesis is the process of creating blood cellular components in the blood marrow. Neutrophils and monocytes are derived from a common myeloid progenitor cell. Monocytes are going to travel in the blood, and when they get into a tissue, they will differentiate into macrophages and some dendritic cells. B cells, macrophages, and dendritic cell are all classified as antigen-presenting cells. Antigen-presenting cells engulf microbes and process the antigens. After this, they will present those antigens via MHC class 1 and or MHC class 2, depending on the cell. This will lead to the activation of T cells. Dendritic cells are derived mainly from monocytes. They are the bridge between the innate and the adaptive immune response because they process antigen material and present it in the context of MHC class 1 and MHC class 2. Found on their surface. They present these antigens via the MHC molecules to the T cells. Dendritic cells activate CD4 positive T cells by presenting pathogen peptides in the context of MHC class 2. Similarly, dendritic cells activate CD8 positive T cells by presenting pathogen peptides in the context of MHC class 1. Upon an injury, such as a skin injury, dendritic cells take up microbes in the tissue via phagocytosis. Then they move through a draining afferent lymphatic vessel. Dendritic cells bearing antigen enter the lymph node and settle in the T cell areas, or the paracortex. Then they present peptides of the pathogen in the context of MHC1 or MHC2 to activate CD8 and CD4 T cells of the adaptive immune response in the lymph node. Some activated CD4 T cells, T helper 2 cells, provide help to B cells to produce antibodies. And other T cells, T helper 1 cells, help CD8 T cells and natural killer cells. CD8 T cells will leave the lymph node and go toward the infected tissue to control viral infections and intracellular pathogens. Effector CD4 positive T cells will leave the lymph node and go to the infected tissue to activate macrophages and control the infection. Macrophages are cells specialized to reside in specific tissues. Monocytes differentiate into macrophages in tissue. The main marker for macrophages is CD14. Some of the functions of macrophages include the detection or recognition of pathogens via PRR, pattern recognition receptors such as Toll-like receptors or TLR, and CD14, which recognizes LPS found on gram-negative bacteria. Phagocytosis is involved in the killing of pathogens. Macrophages use the phagolysosome, which is a fusion of a phagosome and a lysosome, and they secrete antimicrobial products such as reactive oxygen species, or ROS, for extracellular killing. Furthermore, macrophages produce pro-inflammatory molecules. For example, chemokines such as CXL8 or interleukin-8, and cytokines such as interleukin-1, 6, 12, and tumor necrosis alpha. These molecules lead to the activation of endothelial cells and the recruitment of neutrophils. Macrophages are also involved in tissue repair, and they play a role in adaptive immunity. As mentioned before, along with B cells and dendritic cells, macrophages are antigen-presenting cells, meaning that they can present antigens to T cells. Cytokines produced by macrophages, more specifically interleukin-12, induce T cells to become T helper 1 cells, which are a subset of CD4 positive T cells. Neutrophils are polymorphonuclear leukocytes, or PMNs. Other polymorphonuclear leukocytes include the non-phagocytic eosinophils and basophils, to be discussed in another video. CD66 is the membrane marker for neutrophils. Neutrophils are the most abundant cells in the blood, representing more than 50%. They kill mainly extracellular bacteria. In addition, neutrophils contain enzyme-rich lysosomes and granules. These release lysozyme, defensins, myeloperoxidase, elastase, and many other effectors. Specific or secondary granules include NADPH oxidase and lactoferrin. Lysosomes are also present in the neutrophils. They contain an acidic pH, as well as lipases, proteases, and nucleases that will be used in the killing of microbes. Neutrophils recognize pathogens via multiple PRRs, such as mannose, glycan, TLR, and FMLP. They are short-lived cells with a lifespan of about 2 hours. Neutrophils are responsible for post-formation seen in pyogenic infections. After watching this video, please rate it by using the thumb up or down option below. Thank you.